So we've just talked about genetic variance, but that is only one, or you could argue two, genetic and variance. Those are only two of the requirements of evolution. The last requirement for evolution to take place is some sort of mechanism of change. How is that genetic variance changing? In this video, we're not going to talk about all of the mechanisms of change, but instead focusing on gene flow, genetic drift, bottleneck effect, founder effect, and non-random mating. So you've seen this before, but just as a quick recap, again, evolution is just the change in that genetic variation in a population over time. And we've already covered the first two requirements. One, there has to be variation in that population, and that variation has to have a genetic basis. So for example, in humans have different hair length, so that's variance, but that's not a genetic factor. The last requirement is a mechanism of change. Cool, you've got genetic variance, but evolution's not happening unless that genetic variance is changing over time. And there's lots of ways that our genetic variance can change, and that's what we're going to explore next. So the first way our genetic variance can change in a population is through a process called gene flow. Now, in real life, what this looks like, you're not actually seeing genes moving from one population to another, but you do see individuals that are immigrating and emigrating. Organisms don't exist in just one large megapopulation, but instead most of them are found in clusters. And within those clusters, there might be a unique genetic variance of that population. But as organisms move from one population to another, they may have the potential to be introducing new genes or new combinations of alleles that population hasn't seen before. So here I have two examples. On the left-hand side is a lion's pride. In Africa and in other areas. So in Africa, when you have a lion's pride, it's not the only one, right? There's lots of different pride of lions. And so what we see is, not to go into like the super nitty-gritty of it, but most lion's prides have one male and then multiple females. And when new male cubs are born, they grow up before they end up leaving that pride and then go to start their own, look for a new population, etc. Well, that initial pride had a unique set of characteristics that may or may not be shared with other prides. So when that male leaves and goes elsewhere, there's the potential that they might be introducing new alleles, or again, new combinations of alleles with those that they reproduce with in a new population. This second picture, it's really easy to focus on the bee, but the bee is not the example of gene flow here, but it's actually the flowers the bee is on. It's really easy to think of evolution when we talk about animals, but keep in mind lots of groups of organisms, plants, fungi, protists, bacteria, are evolving as well. Now with plants, you could think of a field of wildflowers, and then miles and miles away there might be another field of wildflowers. And while genetically they're probably pretty similar, because they're isolated to one area, they likely have, again, there's similar genetic makeup of that area. Well, bees and other pollinators, what they do is they're moving pollen. Pollen is some of the reproductive material in plants, moves that pollen from one group of organisms, and then maybe that bee flies a couple miles to another field of flowers and drops off, so to speak, that pollen or that reproductive material. And so here what we mean by gene flow is that the genetic material from one population is mixing with a new population. And with that, you might see a change in the genetic variance depending on what those new individuals looked like or what their alleles were. So this is one way that the genetic structure of a population may change, is through the gaining or losing of some of its members. Another way that our genetic variance may change, or the allele frequency may change, is a phenomenon referred to as genetic drift. And overall, genetic drift just means, yes, the allele frequency has changed, but it's by chance. Like nothing particular is going on. So it's not individuals leaving and coming. It's not because some of them could get more food. It's not because some of them were more attractive. It's not because not because of anything. It's literally just random chance why the allele frequencies were changing. You can almost think of this as like a right place, right time. And what I'm referring to there is thinking about the individuals that reproduce. So let me give you an example. In this picture, we have 10 rabbits. So we have a small population of 10 rabbits. And this is showing you their genetic makeup. So we've got an even number of homozygous dominant and recessive, and an in-between number of our heterozygotes. Now, what I mean by right place, right time is the ones in this red circle is representing, you know what, in this generation, those are the ones that happen to reproduce. Not because for any particular reason, not because they were more attractive, it wasn't because they survived longer, it's just those were the ones that happened to reproduce. No reason behind it. So if those 5 are reproducing with one another, here's our next generation. Here's what our next generation looks like. We're starting to see our allele frequencies change. We're having a lot more big Bs, big B alleles in this population, and a lot less little b alleles. Let's do another generation. So again, no reason at all. It was just those 2 rabbits that happened to breed that generation. Both of them being homozygous dominant, which means our subsequent generation is 100% homozygous dominant. Evolution just happened, right? We just had a change in allele frequencies. This image shows us, hey, the allele frequency originally was 50-50, and now it's 100% big B and 0% little b. Now you may be looking at this and just see P and Q. We haven't learned about this yet, but we will explore that more. But just know we're looking at big B and little b. So we had a change in allele frequencies, but it wasn't for any particular reason. It's just those were the ones that happen to reproduce. That's it. So genetic drift, it seems kind of like, I don't know, it almost feels wishy-washy because there's no reason for it, but it's just essentially keeping in mind like random chance exists, and this is the official name of random chance. Now, genetic drift can actually be kind of broken up into 2, I guess you would call them subcategories, so let's explore those. The first one we're going to explore is bottleneck effect. And so bottleneck effect is still a type of genetic drift. So bottleneck effect is still random chance, but I'm trying to think of the best way to word this. But the reason it's random chance is related to surviving individuals. This is going to make more sense as I talk about the example. So the term bottleneck effect kind of comes from this image or this mental image that you might have. So imagine this bottle is a population, and all the beads inside of it are representing a population. There's different colors just to kind of represent the different genetic makeup of each one. So if they're similar colors, they have similar genetic variants. If they're different colors, different genetic variants. Now all of this population's in this bottle, but you see this bottle is being poured into a cup. And if you look at the bottleneck, hence the name bottleneck effect, the bottleneck represents some sort of event that is going to affect the survival of those species. You see it narrows down, so only a few individuals are making it past that event. Our new and our surviving population is a whole lot smaller. But the reason the surviving organisms are there is random. It wasn't because they were able to escape that event. It wasn't because they were faster or could get more food or anything like that. It was just random chance that those were the ones that survived. Well, if you had a really large population and you now went to a really small population, the organisms that are in that surviving population, there's not as much genetic variance. There's just not as many individuals. And so you're going to see that allele frequency change drastically. To give you a real life example, cheetahs are a beautiful example of a bottlenecking effect. So cheetahs have been around a long time, like very long time. It's sometimes hard for us to think of like, oh, like they've been around forever. So their population, I don't know the actual time scale of them, been around for quite a while. But the bottlenecking event that happened was our last ice age. Our last ice age was about 10,000 years ago. Now, cheetahs existed on Earth during this time, but the Ice Age really hurt their population. And scientists believe it was somewhere between 8 to 12 cheetahs that survived worldwide. So not a lot at all. And this was kind of crazy because the reason they survived wasn't because they had thicker coats or they were able to dig better to get more food or anything like that. It was just they happened to be at the right place at the right time that ensured their survival. There was no actual characteristic that helped them to survive. Well, if you only have 8 to 12 cheetahs left, the amount of genetic variation that is going to exist is not a lot. And when the only organisms they can reproduce with is one another, and there's only 8 to 12 left, you can imagine that there's not a whole bunch of genetic variants being introduced into this population, and so you have a very, very small genetic variance. This has actually been detrimental to the conservation of cheetahs, which not only were impacted by the Ice Age, but also in present day, thinking about humans and poaching, is that breeding efforts of cheetahs is so poor. Zoos around the world are trying to breed cheetahs, but because of so much inbreeding, because again, only 8 to 12 individuals, because of so much inbreeding, they have a lot of issues with reproduction. And a huge fear is that if one cheetah were to be susceptible to, say, a new disease or a new fungus or new parasite, they're likely all to be susceptible. Cheetahs between individuals are about 99% identical to each other. They're essentially just as identical to each other as identical twins. Every single cheetah in the world. Meaning if one's susceptible to something, they're all susceptible to that. So that was a little bit of a tangent about cheetahs, but as a recap, again, the bottleneck effect is just a type of genetic drift. You still have random individuals that are reproducing, but in the case of bottleneck effect, the random individuals reproducing were survivors after an event. So that event could be worldwide, like an ice age, or it could be something like a hurricane, or it can be a wildfire. So some sort of event is killing a bunch of individuals, only leaving a few behind that are reproducing. Another example of genetic drift is the founder effect, and it's kind of working similar-ish to the bottleneck effect. So similar to the bottleneck effect, similar to our definition of genetic drift, you have a large population, but only a small subset are reproducing. With the founder effect, the way this works is you have a large population, and then the small population leaves. And essentially what we're looking at is that small population that's leaving. The small population is going somewhere else and founding a new area, and they're reproducing with one another. Well, if you're a small population, I mean, you're just breeding with one another, and so the genetic variance is going to be limited, but again, different. It's going to change from that large parent or original population. Now, in order for the founder effect to really be seen, in order for that genetic variance to start changing significantly from that parent population, there's got to be a barrier that separates it. If the organisms that left keep going back to the main population, you're not going to see evolution happening because those genes still have a chance to transfer back and forth between the populations. The founder effect, you can think of it as a group leaves and isolates themselves. Humans, or certain human populations, are great examples of this. So I'm not really familiar, or I'm not sure how familiar you are about the Afrikaners population in South Africa, but the Afrikaners is actually a subset of the Dutch population. So talking about humans, Dutch being the Netherlands. So hundreds of years ago, back when we as in humans were like actively colonizing everything. Here we have the Netherlands. They have a large population, lots of genetic variants, lots of people, but then a small subset of them, essentially colonists, went down to South Africa. So literally very large distance. That's what their barrier is. It's just all of this land. Go down to South Africa just to establish there. And what's really interesting about the Afrikaners population, so Afrikaners referring to this group of people from the Netherlands, is that just a couple, it was either 1 or 2 individuals had Huntington's disease. Now, in the human population, Huntington's disease exists, but the incidence, and this number is going to be off, but the incidence is like 1 in, 10,000. But in this population of 100 or so people who went down to South Africa, 1 or 2 of them, and we'll be conservative, we'll just say 1 of them had Huntington's disease. So in the main population, you had incidence of 1 in 10,000, and in this small little founding population, you had 1 in 100. Already we're seeing a change in the allele frequencies. Huntington's disease is actually a dominant disease, and so it was actually able to spread throughout that population. Now, when I say spread, I don't want you to think like, oh my God, like every other person had Huntington's. It's not like that at all. But it's still spreading, and it's causing a higher incidence of Huntington's disease in this small little population where they're only breeding with one another versus that large original population. Now, this particular case study is looking at just Huntington's, but the idea is still the same whether we're looking at other diseases or just alleles in general. When you're in a really large population, but then only a small number of you are reproducing, in this case, it's a new population that's a lot smaller, your genetic variance is going to be limited. The less individuals you have, the less genetic variance that there is. So we're going to see a change in the allele frequency, and we do call that evolution. No, we're not making a new species. We're just talking about the basic definition of evolution, a change in allele frequency in a population over time. So again, founder effect being a subset of genetic drift, it was random who were the people leaving the Netherlands and going to this new location. It was random who was reproducing in that case. It's just, in this particular case, we're talking about a small subset of the population leaving a larger population. The last type of mechanism of change for our allele frequencies we'll talk about is non-random mating. So this is a little bit easier to describe if I use the term random mating. So random mating would mean me as a human, I have an equal likelihood of reproducing with anybody in the world. If you just pause right there, you're like, well, no, you don't. Right, I don't, right? I'm not going to be like, oh, let me spin this globe and put my finger down and choose someone who I'm going to make babies with. No. Organisms, not just humans, other organisms, plants, protists, bacteria, fungi, most organisms undergo non-random mating, that you're choosing a partner for a reason. Now, the most common reason, and the most maybe easiest to explain reason, is physical location. So thinking about humans, most of the time, we're choosing a partner geographically close to us. Now, you might be like, well, I also choose them because X, Y, Z. You totally do. Most of that falls under another category, but just think about physical location. You're probably going to be with someone who literally is close to you. You're not going to be like, you know what? Greenland's looking pretty cool this year. Let's go find a mate. Let's go find a baby daddy over in Greenland. Like, you're not going to do that. And that's not just with humans too. I mean, think about just animals in general. Like, you don't hear the story of like, yes, the gray wolf in Maryland goes to California every year to reproduce with his mate. I don't know. The point being is this one is a pretty big factor when thinking about evolution. Humans is a beautiful example of this because think about, and this sounds bad to say this, but think about the stereotypes of different races of humans, ethnicities of humans, but just different countries. If you look at someone, especially someone whose roots have been in that country for a while, so for example, look at a Chinese person versus a German person, and they've been living in Germany, and their family's been living in Germany their entire lives, and they've been living in China and grew up in China, and their family's been in China their entire life. You see physical differences between them, and I don't think any of you can argue with me about that. And those physical differences is due to non-random mating. The reason we're able to identify a German person versus a Chinese person is because over multiple generations, people in that geographic area were reproducing with one another. People in that other geographical region were reproducing with one another. So the alleles become unique to that area, and if they keep reproducing with one another, they stay in that area and become more and more unique. This is actually how a lot of the Ancestry and 23andMe and things like that work, is that there are unique allele combinations for different ethnicities and cultures around the world. And that's how those Ancestries ones work, because they look at your alleles and they're like, oh, well, that's a pretty unique Brazilian root. Yeah, so people is a great example, but this applies to other living organisms as well. So again, when looking at evolution, you have to have that genetic variance. You also need a mechanism to change that genetic variance. So gene flow is thinking about immigration and emigration of alleles, of individuals that might have different combinations of alleles. Genetic drift is just who ends up reproducing is random, and that can impact that allele and gene frequency. The bottleneck and founder effect are two examples of that genetic drift. And then finally, non-random mating is that organisms typically are choosing other organisms that are physically close to them. And so that's going to start creating a unique gene profile that's going to be different than other physical locations. Our next video is going to go through the final mechanisms of change, the ones that you might be more familiar with, such as natural selection.